All right, we are now going to talk about DNA extraction from cells. This will set up the information that you need for a lab that we'll do later this week. The lab that you're going to do is called DNA extraction, and you're going to take samples of DNA from your cheek cells as well as from strawberry cells, and we'll compare what they look like. So I thought that it would be a good idea for you to understand a little bit about the size and scope of DNA itself. So if we were to look at one human chromosome, just a single chromosome, it's about 0.05 meters long. Okay, so it doesn't sound like it's very long, but remember, this is just one chromosome, and in humans, we have 46. So if you have 46 chromosomes, and each of them basically measures 0.05 meters long, then the entire DNA molecule, when you link them up from chromosome 1 all the way to your sex chromosomes, will be about 2.3 meters long. And remember, this is from one cell, okay, just a single cell. So the DNA from a single cell is over 6 feet long, okay? So just how many cells are there in the human body? Well, you have a whole variety of cells. You have brain cells, fat cells, muscle cells, blood cells, nerve cells, okay? When we make an estimate, okay, we estimate that there's approximately 1.0 times 10 to the 13 cells in the average adult human. That's a lot of cells, okay? So if we take that particular number and we multiplied it by the length of a single DNA molecule from a single cell in the body, that now becomes 2.3 times 10 to the 13 meters of DNA that is present inside the average person. Okay, so again, the number itself might not seem to have changed very much, but 10 to the 13 is a very large number. Okay, and to show you how large this number is, okay, the length of DNA in an average person is therefore 23 billion kilometers. Okay, and 23 billion kilometers is a very large length of distance. Okay, so large that we actually need to look at the solar system in terms of comparison. So the distance from the Earth to the Sun, okay, the Earth is right here, third planet from the Sun. It's only 149.6 million kilometers. When you look at the radius of our solar system, if our solar system stops at Neptune, okay, so we're not really including Pluto or the Kuiper Belt materials there, right? So if it stopped with the planet Neptune, then our solar system is roughly 4.5 billion kilometers in length. 4.5 billion kilometers is still a very short distance compared to 23 billion kilometers. Okay, in terms of how far 23 billion kilometers is, humans, the very, very first probe that we sent into deep space was called Voyager 1. And Voyager 1 is currently sitting at 21.7 billion kilometers away from Earth. So if you were able to stretch a person's DNA, okay, all the way throughout our solar system, it will go beyond our solar system, and not even Voyager 1 would have reached that distance yet. So that is one very long set of DNA that can be found inside a person. And all that DNA has been coiled up so tightly that it basically fits inside all the cells in your body. So that's mind-blowing, actually. So how do we get it out? Well, there's basically 4 or 5 steps to this. So I'll kind of go through the process here right now so that you have a general idea. And then I'll go through the process again when we're actually in the lab. Okay, and I'll actually show you a few techniques in terms of how to extract your DNA without damaging it so much. So one of the first things that we need to do is to obtain a cell sample. So like I said, we'll likely take some cheek cells and we'll likely take some strawberry cells. You can pretty much use any type of vegetable. Okay, those are easy for us to obtain the cells from. Okay. You're going to need to find a way to destroy the cell membranes. Okay, because remember, your DNA is found inside the nucleus. So destroying the cell membrane allows you to access the nucleus, but once you have the nucleus, you still need to be able to get inside the nucleus because it is also protected by a membrane. And the way that we do that is by adding something called an extraction buffer. This extraction buffer is simply a mixture of salts and soap, like detergent, like dishwashing detergent. Once we've destroyed the cell membranes, we would have released the DNA. But all your genetic material is kind of clumped together still. Because remember, all that DNA is wrapped around histone proteins. So we need something to destroy the proteins. So we're going to add an enzyme called protease. And protease will destroy those proteins. And once those proteins are destroyed, all we're left with, in theory, is your DNA. Okay. But the DNA won't be visible to your eyes. Okay, so we're going to have to find a way to make it visible. Okay, so in the chemistry world, we want to precipitate the DNA. And to do that, we'll have to add some alcohol, cold alcohol to be more accurate. So this DNA solution that you have that contains basically cells, broken cell membranes, destroyed histones, okay? You need to separate all that stuff from DNA, and alcohol will help to do this. So once that is done, you're going to collect your DNA and you're going to admire how wonderful that DNA is. So hopefully you'll get a chance to do that sometime this week.